Hi, everyone, and welcome. My name is Caroline with Fiveable. Happy 2019. It's been a while, so welcome back, or thank you for joining if you haven't joined before. And tonight, as you can probably see, we're talking about Mendelian genetics. So I'll go through all the basics of genetics, and this is going to be used because we'll build off of this. So next week we'll talk about chi-square analysis of Mendelian genetics, which is a really, really important episode. So if you know people who are in AP Bio, definitely tell them to stop by because that's going to be a big one. But you have to know the basis of genetics really, really well before we can jump in and add on. Because I don't know where you're all coming from and if you've had an experience with a biology class before, I have what I think are all the basics on those slides. If I'm going too fast or too slow at any time, just let me know. But feel free to ask lots and lots of questions if I, kind of zooming through something and you're not sure what a word means or anything like that, just because it's hard for me always to know what you do and don't have experience with based on what classes you've taken before. Okay? All right, so we're going to jump into the PowerPoint. I don't know if we'll be on for a full hour tonight. I guess we'll see. It's a really good practice problems, and then next week's going to be a really big week for us. It'll be a long week because we'll be talking about Okay. So a little bit of background here so we understand what we're talking about. So Mendelian genetics, when I say that, if you've ever learned about genetics before, you've most likely learned Mendelian genetics, which is like the first type of genetics that we learned about as a human species, and it's the most basic. Unfortunately, most things don't actually fit Mendel's rules. But it's still really important to know what things do and how these rules work because it set up the foundation for all of our progress with genetics that we have today. So this is a lovely little picture of Gregor Mendel. He was a monk living, I believe, in the 1800s. I should fact check myself though. And basically he would do scientific experiments on pea plants at the monastery. And what he was finding was, and he started calculating all of this data, that there was a correlation between the parent pea plants and then the offspring. Now, this sounds not too earth-shattering, but you also have to realize that when Mendel was doing this research, we still did not know that DNA was our genetic material. We didn't even really know if genetic material existed. Okay, so Mendel really set down the foundation of like, hey, there's genetic material and it's passed down from generation to generation, which was earth-shattering. Okay, then he also started finding out things like alleles and kind of how those, well, not quite yet. I guess I jumped ahead of myself. But he started figuring out patterns, I guess I should say, of inheritance and set some really important foundations for when we actually did learn about DNA and alleles and all of that good stuff. So what Mendel was kind of seeing here is he was doing some tests and he would basically transfer and pollinate plants with little paintbrushes you can see here. And so he did one cross where he crossed a purple plant with a white pea plant as far as their flower colors go. And the first filial generation, which we call F1, and I have this all actually in the bottom of the slide. If you look at the speaker notes, oops, that's what I'm going to do. Down here, I have some vocab. I tried to make it a little easier this week instead of typing into the chat box where it can disappear. So if you are referencing and you aren't in presentation mode, that's helpful for you. So we have this parental generation, which we always call the P generation. That's our first cross. And then when we cross that parental generation, we make the first filial generation, which we call the F1 generation. And he noticed that when he crossed a purple plant and a white plant, he saw all purple plants. And this was like earth-shattering. What happened to the white? Where did it go? How did this happen? Okay. So then he takes it a step further. He still does the same starting cross, the same P generation cross with purple and white flowers, leading to that all purple F1 generation. Now he takes it a step further. And he cross-pollinates some F1 plants, those purple plants that are right here, and makes this F2 or filial 2 generation. And this is where his mind was just blown because all of a sudden the white trait came back. Okay, which was crazy. So we had about 705 purple flowers and 224 white flowers. And that was a crazy phenomenon because this white trait, he described it as going dormant and then reappearing, okay? And these types of crosses that he was doing, where he was counting up all the different offspring of various test crosses that he had with his pea plants in this huge garden, really led to our foundation of understanding how things are inherited and why. So this was some really big news. You'll notice that the only differences kind of here between these two experiences is that he took a step further and crossed two of these F1 generations to make a third generation called F2, in which that trait then reappears. And we'll figure out, if you don't know already, we'll figure out why that trait reappears as we start talking here, okay? All right. Okay. So a little bit of background, and again, if you look at the speaker notes here, you'll see a couple vocab words that you need to be familiar with, okay? But Mendel did these crosses for a vast number of things, as you can see. Flower color, seed shape, pod color, flower position, and stem length. So these are the 7 characters, is what they're called. Characters, or I like to think of it as like a characteristic of the pea plants that he was looking at. And each of those characters or characteristics had 2 possible traits, which he deemed dominant and recessive based on what showed up in the F1 cross, okay? So what we see here is we cross a purple and a white flower. We have all purple flowers. So he's like, oh, okay, so purple is the dominant trait. Purple overshadows this white trait. And then it's not until we get our third generation or our second filial generation that we see that recessive trait being able to show its head again. So we'll talk dominant and recessive a lot there, but that's the easiest way to tell. And sometimes in an AP problem, they'll just tell you we have a true breeding, which is going to mean homozygous, and we'll talk about that in a second. A true breeding yellow plant and a true breeding blue plant are crossed, and all the offspring are blue. And you would have to know that that means that the blue plant is therefore the dominant one based on that. Okay, so these guys, again, are called characters: flower color, seed color, those general characteristics. And the traits are the actual like version of that character, I guess you could say. So like yellow and green, round and wrinkled, inflated, constricted, etc. And I have some more examples in the notes at the bottom of the PowerPoint that you can take a look at in case you get confused. But those vocab words are going to be really important in your study of genetics. Okay. So Mendel kind of lays this foundation, right? He figures out what dominant and recessive means. He realizes that these traits can go away and then come back. He's able to get a lot of useful data on like the ratios between dominant and recessive traits. I'm so sorry. 2019, getting back into it. If this was your first week back at school, I feel you. This is my first week back, and it is tough. Okay, so he finds out all this stuff. It's going to lead us to the foundation of Punnett squares, as I said, but we still have a lot to learn, okay? So after him is when we really figure out that DNA is indeed our genetic material, because again, that was not something that was widely known. It wasn't known prior to like the early 1900s, which is crazy to me, like 100 years ago. And then we also figure out that there's a specific location on the DNA, right? So if you think back to all of our videos on DNA and genes and converting genes to proteins, we've talked about this a good amount. We also talked about it when we talked about Hardy-Weinberg equilibrium in evolution. But we figure out that alleles exist, okay? And basically, what this means is, so we have these pairs of homologous chromosomes, okay? And really important, if you weren't here the week, not the week right before break, because that was a Kahoot game, but like middle December episode on mitosis, meiosis, definitely check it out if you're confused about the language that I'm using right now. But we have this pair of homologous chromosomes, which means one's from mom, one's from dad, okay? But the genes are going to be the same type of gene along, or coding for the same characteristics along the chromosome. Okay, so in this location lives the locus for the flower color gene. So the character of flower color exists on this chromosome in this location in every single plant. Okay, and we call an allele a specific version that codes for a specific trait, either the dominant trait or the recessive trait. So in this individual, they have one allele for the dominant trait, purple flowers, and one allele for the recessive trait, white flowers. So we'll see this in a slide, but this individual would be considered heterozygous, hetero meaning different, okay? And because they have one version of the dominant and one version of the recessive allele on their homologous chromosomes, okay? Okay, so that leads us to this kind of basic definitions of various important concepts in genetics, okay? So with this, what we eventually figure out and are able to conclude is that individuals with these two alleles for every single characteristic, they can either be homozygous dominant, they can be heterozygous, or they can be homozygous recessive, okay? And basically what that means is if you're homozygous dominant for a trait, homo means same, okay? And so homozygous means same alleles, dominant, right? Homozygous dominant. So they have two dominant alleles. So their homologous chromosomes would have two alleles for purple instead of one for purple and one for white. So both of these would code for purple flowers. They're considered homozygous dominant. Our middle individuals here are heterozygous, which is what we just talked about with the actual example from the previous slide. Hetero meaning, again, different. So they have one capital dominant allele. We always show that with a capital letter, and one lowercase recessive allele for this specific character. And so this picture would actually be representative of a heterozygote, as I already said, with one allele for purple and one allele from white. They have that mix. Okay. Lastly, homozygous recessive, same general concept, homo meaning same, recessive meaning the recessive alleles. So they have two versions of the recessive allele instead of one or the other or both dominants, okay? So what this also looks like, if you don't have very much experience with genetics, if you have at least one dominant allele, right, you are going to show the dominant trait. You are going to look dominant, okay? And that idea of showing or looking or appearing dominant is called or recessive, is called a phenotype. And I always remember this as phenotype is a physical appearance. It's not always physical, right? Like we can't always see it necessarily. Like sometimes it's something that's going on internally in your body, but it's the way that something would appear based on what the genes are telling us. And so in this case, the phenotype options are either purple, you look purple, which would be the dominant phenotype, right? And we figured that out based on who kind of won out in Mendel's crosses, okay? And then the white coloring would be the recessive phenotype, okay? So in this little example over here, we only have two phenotypes, dominant purple and white recessive, okay? And that's the way that something appears. As long as you have at least one dominant allele, you are the dominant phenotype, okay? And this is going to be so important when we take a look at genetics and even when we go back and review Hardy-Weinberg, okay? Because if a problem ever just tells you like 70% of flowers are purple, you actually don't know much of anything about those flowers because they could be homozygous dominant or heterozygous. So for instance, having a widow's peak, which is the point at the top of your hairline, if you don't have like a perfectly straight hairline, like I have a point right here, that's a dominant trait. I know I have a widow's peak, so I have the dominant phenotype. I have no idea what my genotype is though, which is gonna be whether I'm actually homozygous dominant, heterozygous, or homozygous recessive, right? Homozygous recessive, but I don't know if I'm heterozygous or homozygous dominant. Could I figure it out? Potentially, if I went back and asked all of my relatives if they had widow's peaks or not, I could figure it out. But for the most part, if they give you numbers like that, it's going to be really hard to analyze them because we have this kind of ambiguous combination of dominant and homozygous dominant and heterozygous individuals that both appear to the human eye to be the same because they both appear to have the dominant phenotype. okay? That's what makes the recessive phenotype so awesome, because we know a lot about that organism if it looks recessive, right? Because in order to have the recessive phenotype, you have to be homozygous recessive, okay? Because otherwise, if you were heterozygous, you would show the dominant phenotype. So you have to be homozygous recessive in order to show the recessive phenotype, okay? So phenotype is going to be that physical appearance. In this example, it would be purple and white, dominant and recessive. For genotype, that's your genetic component here. So that's where you would say homozygous dominant, heterozygous, or homozygous recessive. So there's 3 different possible genotypes in this scenario, but only 2 possible phenotypes, dominant and recessive versus homozygous dominant, heterozygous, and homozygous. Aditi has a question. Sorry, I don't know when you asked this, but I'll answer it now. So are the traits alleles since they express the physical characteristics of the plant? Oh, I answered it. Okay, cool. Awesome. Glad to hear that. Okay, how are we doing so far? Okay, cool. Looks like we're okay. So, to review, we figured out what characters and traits are. We figured out what dominant and recessive is, and now we have a lot of vocab that we can use, like phenotype and genotype. Okay, this all comes into play then, and one of the really important things that we can do here is figure out results of 2 individuals mating. Okay, we would call that a parental cross. 2 parents crossing or intersecting their genes. Okay, and that's going to help us to figure out what the offspring of those individuals might look like or could look like with some sort of number attached to it. Okay, so this probably looks familiar to you. You've probably done a Punnett square before. But what we do here is we set the 2 genotypes. So these are 2 heterozygous parents, one capital A, one lowercase a for some random trait. Okay, we set the 2 genotypes on the side and top of our Punnett square, and then we're going to work to fill in the 4 possible offspring that could be created from these 2 parents. Now, why do we have these genotypes separated, right? We have like an A at the top of one column, and a capital A at the top of one column, a lowercase a at the top of another, etc. We have them kind of split into their own rows and columns. And the reason we're doing that is because of independent assortment, okay? So again, if you're unfamiliar with this, tune back into the episode on mitosis and meiosis from before winter break. Basically, when we are dividing up our genes, we are actually splitting our alleles, right? And so that capital A from dad may go in one egg or sperm, and that lowercase a from mom would go in a different egg or sperm, but they would be separated from one another. Because remember, when we make egg or sperm, those are haploid cells that have half the genetic material of a whole human. So they only have one version of each trait, or sorry, each character in each egg or sperm. So they would just have one allele for each trait in there. So that's why we split this over here and this over here, because when this individual is reproducing, half of their offspring would get the dominant A and the other half would get the recessive A based on this splitting. They're capable of passing on either the dominant allele or the recessive allele to their offspring. And when you combine that with another heterozygote, we'll take a look at what those results mean. Okay? All right. So when we're going through this, I always think of it like battleship. And if you're familiar with this, this is just total review. But anytime you have a row and a column intersect, those 2 letters would stick next to one another, and that creates a new genotype. So obviously this alone is not a genotype, but when we have these 2 capital A's coming together, we have a full-fledged offspring with the genotype capital A, homozygous dominant. Okay, we can continue to fill this out, and you'll see we have capital A, lowercase a in our upper right-hand corner, capital A, lowercase a in the lower left-hand corner, and then lowercase a, 2 recessive alleles in the bottom right-hand corner. Okay, you'll notice that here, when we have this blend of dominant and recessive, the dominant letter will always go first. That just makes sure that that you can keep things straight in your brain. It's just a good overall convention to use. Okay, so that's what's going on there. And then we can actually analyze these results by looking at the phenotypic ratio and the genotypic ratio of the expected offspring for these two individuals. So what that looks like, the phenotype, remember, that's how an individual actually looks, okay? Remember that for these monohybrid crosses, and we call them monohybrid because one, mono meaning one, we're crossing one single character here, okay? With a monohybrid cross, there's only going to be two possible phenotypes. These individuals can look dominant or they can look recessive, okay? Remember that homozygous dominant and heterozygous individuals both appear to the human eye to be dominant, okay? So of these four predicted offspring, 1, 2, 3 of them, 3 out of 4, are predicted to have a dominant phenotype, okay? The other 1 out of 4 is predicted to have the recessive phenotype with that lowercase a in the bottom right-hand corner. That homozygous recessive individual will look recessive, okay? So basically what this means is, and I'm going to talk about this on the next slide as well, if these two individuals reproduce, every time they reproduce to make an offspring, there is a 75% chance, a 3 out of 4 chance, that the offspring will have the dominant phenotype for whatever character we're looking at. And there's a 25% chance, or a 1 in 4 chance, that their offspring will have a recessive phenotype. That does not mean, right, if they have 4 kids, that 3 of them will be dominant and 1 will be recessive, right? It just means the odds show that. So, but each time they have a child, there's a 3/4 chance that it's going to be dominant and a 1/4 chance that it's going to be recessive, okay? It's a really important distinction. Sometimes students get confused because I'll be like, all right, an individual has 6 kids, and they're like, oh my gosh, but there's only 4 boxes. And it's like, yeah, but that's just a predicted ratio, okay? So don't get bogged down in that. We can also take a look at genotypic ratio, which would be our 3 genotypes: homozygous dominant, heterozygous, and homozygous recessive. And we can say that 1 out of 4, there's a 1 out of 4 chance, or a 25% chance, that the offspring of these two would be homozygous dominant. There's a 1 in 2 chance, or a 50% chance, that the offspring from these two individuals would be heterozygous. There's a 1 in 4 chance, or a 25% chance, that an offspring from these two individuals would be homozygous recessive. Okay, so easy enough with that. Okay, so this is my reminder to you that Punnett squares are truly just predictions. Okay, so it's just giving you the percent chance that each time that those two individuals reproduce, that their offspring would look one way or the other, have these genes or the other genes, but it's really just a tool of prediction. Okay, and you can practice with these. They're not too challenging if you want to. Okay, in order to be successful with the Punnett square, you really have to be able to write a genotype. Okay, and that's using trait, lots of different symbols in order to create these genotypes. And there's also a few keywords that you need to look for. Okay, so take a look at these 12 practice problems. I'm going to try really quick to copy them into another program so that I can actually let me try this. Shoot, I really wanted that to work, and it didn't. All right, well, since I can't put this on my little writing tool, which is kind of a bummer, we'll use that in a second for some other practice problems. We can just verbally go through this. So number 1, heterozygous for height. I would go up to stem height. Okay, it looks like we're using T's. Heterozygous means you have a mix of both alleles, a dominant and a recessive allele. So the genotype for this individual would be capital T, lowercase t. Number 2, homozygous dominant for seed shape. Seed shape up here, round or wrinkled, we're using R's. Homozygous dominant means the same dominant allele, so 2 capital R's would be the genotype for number 2. I'm going to jump down to question 6. homozygous tall, because again, I don't think people need that much practice with this, but if you need it, you can obviously use it. Homozygous tall, you're wondering why it doesn't say dominant or recessive. It doesn't have to, because I can find stem height tall, and I see that tall is a capital T. So it would be 2 capital T's for the genotype for question 6. For number 7, it doesn't tell us homozygous or heterozygous. It just says constricted pod. But if I look up here in my chart for pod shape constricted, I see that constricted is the recessive allele. So if an organism was to have a constricted pod, it would have to be homozygous recessive, because that's the only genotype that leads to the recessive phenotype. So I would use 2 lowercase s's for number 7. Same would be true for number 8, except it would be 2 lowercase t's this time. And then the last ones that we'll review are 11 and 12. When it says pure and hybrid, pure means homozygous. Hybrid means a mix, just like a hybrid car is a mix of 2 different fuel sources, right? So pure wrinkled would be wrinkled right up here, lowercase r, homozygous recessive. Hybrid tall would be a mix of 2 alleles. We would have the uppercase and lowercase t there for the genotype. You have to get really comfortable with writing genotypes because you're going to use them all the time when you make Punnett square. Okay, so 2 monohybrid cross practices, and I'm going to jump over to my other application in a second, but you guys can keep your PowerPoint open because it'll be helpful. And then we're going to talk a little bit about dihybrid crosses. Okay, so our first practice question is about 2 plants are being crossed. One is heterozygous for seed color, and one is homozygous dominant for seed color, right? And I have a couple questions listed there. So heterozygous for seed color, I'm going to go back here. Seed color is using the letter Y. Okay, so heterozygous would be capital Y, lowercase y. And then homozygous dominant would be 2 capital Y's. Okay, so I'm going to switch the screen that I'm sharing with you quickly. Here we go. Eventually. Apparently me trying to share that screen was enough to crash my Google Chrome. So let's try this again and hope it doesn't crash. Sorry about that. I have the most technical problems of any person alive. Okay, and here we go. Looks like we're getting somewhere. So I can write on this, which is why I'm using it. The problem is I would love to write on my whiteboard all the time, but it's a mirror image, and so everything would look ridiculous, which is why I can't do that. Okay, so we were talking pea plants. We're doing a Punnett square, and our parent genotypes were capital Y, lowercase, homozygous dominant, and then we had a heterozygote parent. So uppercase Y, lowercase y, and I'm just going to make that curly really big so that you know the difference. Another tip here, like if you're using S's or something and you have to make a lowercase s, I always put what I call a little hat over those terms because it's really, really hard to differentiate, especially if you're doing like a dihybrid cross. Okay, so if we're crossing these 2 individuals, okay, these are our genotypes if you're following along with the PowerPoint slide. Mine's pulling back up really quick. Let me just get back to it and make sure I hit all the key points here with you. So the gametes then, gametes are sex cells. So those would be the egg and sperm. So that's what I was talking about when I was talking about independent assortment, how you split the parent genotype, and those would be the possible egg or sperm. Okay, so if this was the male, let's say, the possible gametes from this individual would be capital Y and capital Y. And so we can put that at the top of our Punnett square. Okay, and then the possible gametes from this individual, our heterozygous individual, they can give a lowercase y to their individual or to their offspring, or an uppercase y, or they could give a lowercase y. There we go. Okay, so those are the gametes. And then when we cross, this is easy enough, although this program kind of stinks. Wow, gotta find a better one. We have uppercase y in this upper left-hand box. We have uppercase y in the upper right-hand box. Uppercase, lowercase, uppercase, lowercase, okay? And what results here is, if we were looking for a phenotypic ratio, we would say that 100% or 1 to 0 or 4 to 0, whatever you want, would be dominant, right? Because all 4 of these, even though they're not identical, will show the dominant phenotype, which for seed color is yellow, okay? And the dominant phenotype is usually, so I knew this was seed color and it's a capital Y. Usually the dominant trait is the letter used for the Punnett square. So that's that. So the phenotypic ratio for phenotype, it's going to be like 4 to 0 or 1 to 0. Whereas the genotypic ratio, okay, so you always put dominant to recessive for phenotype. Let's see what this typing thing does. Ooh. Cool. I'm learning. Dominant to recessive for the phenotypic ratio. For genotypic ratio, you're always going to put homozygous dominant to heterozygous to homozygous recessive. Okay, so in this instance, we have 2 homozygous dominant individuals, 2 heterozygous individuals, and 0 homozygous recessive individuals. So 2 to 2 to 0 or 1 to 1 to 0 would be your ratio there. Okay, so easy enough. It's good practice with phenotypic and genotypic ratios. I feel like we're probably good to go on these, so I'm going to go ahead and skip our second example. And we will jump into, I'll switch my screen really quick. Hope my computer doesn't crash. Go. Skip that. We'll jump into dihybrid crosses. Before I do, I think we had a question, so I'll answer that one really quick before we move on to briefly talk about dihybrid crosses. So as far as tips for answering free responses when you're not 100% sure of content, that's a really good question. So one, practice. So ask your teacher for practice free response whenever possible, because there's nothing that can prepare you better than practice. 2, when you're not 100% sure of content, so I always instruct students to skip questions that they don't feel great about. I always tell them to start with question 8 actually and move back to question 1 because question 8 is just going to take you like 1/10 of the time. And I've seen way too many times and talked to way too many readers of the exam to know that students will spend a solid 30 or 40 minutes writing multiple pages for one of the long questions, question 1 or question 2, and get 1 or 2 points for it. So my suggestion to you would be to start with these shorter questions, okay, and then move your way up into longer questions. And you can jump around any section of any question. So oftentimes the long questions will have 4 or 5 sections, A, B, C, D, sometimes E. You can answer question D first. And usually in those longer questions, when you might not be familiar with content, oftentimes there's a section like part D might actually be a math question, or part E might make you write an ethical question. And those are easy points. And when you take a look at the national averages for like questions 1 and 2, it's usually like 2.3, 2.4, sometimes 3 points out of 10. And the national average is what's set to a 3. out of 5 on the AP exam. So if you can kind of work your way through, work on parts that make sense, like a math portion or a question asking portion, and get 2 or 3 points, you're doing just as well as the students who wasted 40 minutes of rambling about something that they're not super comfortable with to also get 2 or 3 points. And then you maximize your time on some of the easier, shorter questions. I always say go back to basics, go back to basic biology, right? You know lots of things about cells from basic biology. You know lots of things about DNA from basic biology. Always include what you know about that topic, even if you think it's far too basic for what the question is actually asking, because oftentimes you can get a point there and it's worth a try. Look for questions that have graphics or tables or graphs that you need to make, because those oftentimes have straight numerical analysis. And yeah, include key vocabulary words when you can, but there's always going to be things that you're not familiar with. So you have to start getting comfortable with feeling uncomfortable about material. You're just going to have to use what you do know and write down some really solid things while also not wasting too much time. So hopefully that helps. We'll do a lot more like specific prep like that closer to the AP exam. Okay, so dihybrid crosses. So now we have dihybrid instead of monohybrid, which means we're looking at 2 different characteristics at once. So in this case with peas, we're looking at both pea color, yellow versus green, which is what we just did that cross for. But we're also looking at seed shape, round versus wrinkled, okay? So we have seed color and we have seed shape here. And when we cross those, we're going to have a couple different results here. But in this case, we have a purely homozygous dominant individual whose gametes will all be uppercase Y, lowercase, or uppercase R, crossed with a purely homozygous recessive individual, lowercase y, lowercase r. So we make all of these heterozygotes. And then when we cross them, we see some really, really crazy ratios. But we're going to try one of these. Hopefully I can get that screen on the next page to work for me. So we're going to try one of these, and I'll teach you guys how to find gametes as well. Okay. So dihybrid crosses are going to have 16 squares as opposed to 4, because we're looking at 2 different characteristics, which is going to yield us 16 possible results as opposed to just 4. Okay. All right. So the question that we're going to be doing is this one, where it says, in pepper plants, green fruit color, capital G, is dominant to red, lowercase g, and round, uppercase R, fruit shape is dominant to square, lowercase r, fruit shape. These 2 genes are located on different chromosomes. What gamete types will be produced by a heterozygous green round plant? Okay, that's where we're going to start. Again, I really wish I could copy this into my whiteboard. I don't know why I can't. I got to work on that. I got to ask somebody. Ooh, wait, maybe I can. Give me a second. Okay, now we're getting somewhere. I think I can maybe do this. Okay, maybe not. Sorry, guys. This is a pain in the butt. All right, well, here we go. Okay, so sorry for making you wait there. I was trying to do something cool with this new program, and alas, story of my life, it doesn't want to work. But I'll show you guys how to make gametes really quick, and then we'll do this cross, and then I'll give you some more practice to do on your own. Okay, so I think I'm making squares right now. There we go. That kind of works out. Let's see if I can cut. Ooh. Okay, I'll try to make us a 16 square box right now. I guess I probably could have done this easier by just going like this and drawing some lines. Oh, you live and you learn, huh? Okay, So here's going to be our dihybrid cross, and we have heterozygote parents. We need to figure out how to make gametes, okay, which remember would be like an egg or sperm cell. So we need one version of each characteristic in that gamete, and then we'll move on from there. Okay, so our individuals, they tell us, are both heterozygous for both traits. Okay, so we have a capital G, lowercase g, capital R, lowercase r parents. So those are the genotypes, okay? And the way that we're going to find all of the possible egg and sperm, we can't just divide them this time, right? Because in each division with each egg or sperm creation, we could have an uppercase or a lowercase g. We could also have an uppercase or a lowercase r. So we're actually going to have 4 possible combinations of these 2 traits in a possible egg or sperm, where we have just one version of g and one version of r, or one version of each characteristic, okay? All right, so I'm going to use a couple different colors here, but really the way that you can find your gametes is through the FOIL method. If you're familiar from math, right, when you're foiling out binomials, that's what we can do here. And our imaginary parentheses will exist between our 2 different characteristics that we're trying to cross between, okay, that we're trying to take a look at. Okay, so the first thing that we'll do to make a gamete is cross the first allele from each characteristic. So we have a capital G and we have a capital R. Okay, so our first possible egg or sperm would be capital G, capital R. Okay, capital G, capital R, capital G, capital R. So I'm putting that at the top and on the side of my Punnett square, only because I know both parents are the same genotype, okay? Next, the first outer O, so our outer alleles are a capital G and a lowercase r. So this individual is also capable of passing on a dominant version of trait 1 and a recessive version of trait 2. Okay? They can also, we have our inside alleles, lowercase g, capital R. So they could also pass on a recessive version of trait 1 and a dominant version of trait 2. And this is crazy for heterozygotes. Heterozygotes are crazy. Okay. Lastly, we have our last terms, okay, and that is lowercase g, lowercase r. So recessive alleles for both traits, okay, and that's how we find the 4 possible alleles, okay. You can always find the 4 possible alleles this way. Sometimes some of those alleles will be the same if the individual is not heterozygous for both traits. Like if they're heterozygous for the first trait, but homozygous recessive for the second trait, then you're going to have some duplicates when you do this method, but it will always work and it will always get you the right answer. So if you don't feel comfortable with coming up with all the possible genotypes, or sorry, gametes from a potential genotype, you can always use the FOIL method to find those gametes. You always need to fact check yourself and make sure that you only have one version of each of the 2 characteristics that you're looking at in each. I've seen students just transfer like capital G, lowercase g, capital R, lowercase r, which is inaccurate because you're only looking at one trait and you need one version of each of those, okay? All right, so now this is a crazy version of FOIL. So I'm gonna actually type here so that we can all save ourselves the trouble, okay? But when we go through this, you're gonna keep the, why does it put that right there? That's super frustrating. You're going to keep the G's and the R's together, okay? So like in my first box, I have capital G, capital G, capital R, because I have a capital G, capital G, capital R, capital R. I keep the G's together. I keep them in front of the R's, and I keep the R's together behind the G's, okay? Next, I have 2 capital G's, a capital R, and a lowercase r. Next, I should have capital G, lowercase g, capital R, capital R. Capital G, lowercase g, capital R, lowercase r. Okay, and hopefully this process makes sense. True. Okay. You'll notice that when something is lowercase, I'm putting it second. Okay, but the one that I just typed out, lowercase g, capital R, even though the G's are lowercases, they still come first because that's our first character in our genotype. So don't feel bad about that. As long as, like in this instance, the capital R comes before the lowercase r. Okay, so it's just amongst the same character that you should be worried about that. Okay, so now we actually have to make something useful out of this. So now that we have all of this, we need to figure out what it actually means. Okay, and so what we're going to do is we're going to go through and code for the three possible phenotypes that, or four possible phenotypes, I'm sorry, I should say, that can come out of this. So these pepper plants can be dominant for both traits. If they're dominant for both traits, they would be green, round pepper plants. Okay. If they're dominant for the first trait, they could be recessive for the second trait, which would make them green and square. I'm getting this from the problem from before, so that's where that's coming from if you're wondering. Okay. They could be recessive for the first trait, but dominant for the next trait, which would make them red and orange. They could also be recessive for both red and square. Okay. And we can figure out the ratio for this by counting up our 16 boxes. Okay, so how I always do that, I usually get like different colors or I use different shapes, and I'll go through and I'll see, okay, which of these, so we'll start with dominant. Which of these has at least one capital G and at least one capital R? Listen to how I say at least one, because we're not looking at genotypes here, so I don't care if this individual is heterozygous or homozygous dominant. They just have to look dominant, right? We're just looking at looks right now, green and round, okay? So this individual is dominant for both traits. So oops, I'm gonna just put my pencil. So I'm just gonna put a pink dot there because that's what I used for my dominant allele. So I know I've counted that one. That one's dominant. Even though it's heterozygous, doesn't matter. Okay, all of these are going to appear dominant for both of my traits. So 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16 are going to be dominant. 9 out of 16. Okay. Now I can count up, I'll use yellow because that was my dominant recessive gamete. I can count up how many are going to be dominant for the first trait, but recessive for the next. Okay, so here I'm looking for at least one capital G and 2 lowercase r's. 1, 2, 3. 3 out of 16 are dominant and then recessive. Okay. Recessive dominant, I need 2 lowercase g's and at least one capital R. Now this is easy because I only have 4 boxes left to count. Okay, 3 out of 16. Okay, and then recessive, only 1. 1 out of 16 peppers would be expected to be red and square. Okay, so this is my phenotypic ratio. You never need to know genotypic ratios of Punnett squares because it's way too crazy to do that. Okay, but phenotypic ratios you do need to be familiar with, and you will be expected to know them. Otherwise, you're going to have to do dihybrid crosses on your AP exam, and you simply don't have time for that. Okay, so dihybrid ratios that you need to know. So that 9:3 to 3:1 ratio that we just created, every time you cross 2 heterozygotes, you will get that. Okay, so here we had those heterozygotes crossed, this F1 generation to make this F2 generation, and we got the 9:3 to 3:1 ratio there too. It will always exist. That will save you a ton of trouble if you realize that's what the parent generation is showing. Then do not do that dihybrid cross. Just know it's going to be a 9:3 to 3:1 ratio. Save yourself a ton of time. Okay, and they kind of expect that you know that, so they may give that to you. Another good one to know is when you have one dihybrid and one homozygous recessive, you get equal distributions of all 4 phenotypes, which is pretty cool. So that's actually the situation with question 2. If you take a look at that in your own time, you would see that the mom is heterozygous for both traits and the dad is homozygous recessive for both. And you get a 1-to-1-to-1-to-1 ratio, all equal of all 4 phenotypes, which is pretty cool. Okay. All right. So that's it for today. On my screen just then. Gosh darn it. I'm sorry. For the last 2 slides of the PowerPoint that I was going over, I'm so sorry. So you can take a look at them on your own time, but it lists those ratios and why they're super important. I'm just like talking to myself over here. And it shows you the other practice problem as well. So those are listed as well as the genotypes that create them, because that's a really important thing to remember. And so those are there for you. Next week's going to be a huge one with chi-square, so carve out some time and get ready to be here. Tell your friends, okay? Otherwise, that's all the time we have. Thanks for joining in, and hopefully I see you next week.